Hello and welcome to the day 4 of IVI Vaccinology course. My name is Sushant Sahasrabudhe. I'm the Associate Director General at IVI. Unfortunately, Zenaida Mojares, who was supposed to moderate this session, she could not join today. So I'm taking on her behalf. So welcome from all parts of the world. I will not say good afternoon, good morning, because we have people who are joining from all parts of the world. So today is going to be again a full session with 5 speakers. 3 of those speakers are actually from International Vaccine Institute, and then we have 2 additional speakers. So we have Dr. Zenaida Mojares, Dr. Thongyong Kim, Dr. Flor Munoz, we have Dr. John Louis Excler, and Dr. Pauline Paterson. So we have a full agenda right now. Before we go to the day 4 agenda, I will have some housekeeping notes. So I think most of these things have been repeated on day 1 to day 3, but I will just, for everybody's understanding, we will repeat this housekeeping notes. So the live sessions are beginning now, and then it will be uploaded on our YouTube channel, which will be available 3 p.m. Eastern time. And these recordings are only available for this week. And the second important point, which we generally get a lot of questions, are that we will please do send out your questions in the Q&A part, and then we will try to answer those questions live Q&A, as well as there will be some speakers who will join us. So we'll try to answer most of those things during the lecture itself. The presentation slides, I think many of you have asked from day 1 to until now about the presentation slides. We will share those slides with you. We will confirm about when we can share those. And the important part for us is in our process to improve this experience for you, we actually want to get the opinion from you about each of those lectures. So at the end of each lecture, you will actually see the polls coming up. Please do participate in that. And the last point is about the certificate. So most of you will get that certificate. The question is, if you attend the 90% of the course from this week, you will get the digital certificate, and we will certainly you have to request to the course secretariat about this request, and we will issue the certificates to you. So now, if we move to the main agenda, we have our first speaker, who is Dr. Zenaida Mojares, who is the Chief Medical Officer at IVI, and she is going to talk on the vaccine clinical development. Zenaida?
Hello, good morning, good afternoon, good evening to everyone. I'm Zenaida Mojares, currently the Chief Medical Officer of International Vaccine Institute. And now I will be sharing to you something about the whole process of vaccine clinical development. So let me present to you a brief summary of what vaccine clinical development is. Now, generally, that the presentation today would include the following topics: a brief run-through of what are preclinical stage development, and then we'll go through the clinical development phase itself. Next, I'll walk you through a brief summary of what is licensure and what are the policies to be followed. Then a brief discussion on who PQ, and a brief discussion also of EUL for the COVID-19 vaccines now. So as you can see, generally, vaccine development undergoes 6 stages, from exploratory, preclinical, and the actual clinical development. Then it goes through regulatory review, and then continuous monitoring of manufacturing and quality control. Now, under normal circumstances, all these 6 stages usually would take 10 to 15 years to get a vaccine into licensure. Now, there are emergency adjustments that can be done to the time frame during emergency situation, as you have seen during this pandemic. But even if you get an expedited approval, all the 6 stages must be conformed with. Now, the 6 stages, I put them in the 6 boxes here that you can see. So first of all, we touch on the exploratory stage. So we also call this the discovery phase, where you identify natural or synthetic antigens in the laboratory. In layman's term, this is where you discover the composition of your vaccine. But before you get into the exploratory stage, of course, you have to undergo some researches on epidemiology to understand the disease that your vaccine is targeting. Now, exploratory phase usually takes 2 to 4 years to complete. And along the way, many vaccines are abandoned simply because it's not successful. I'd like to relate this with the current COVID situation. If you can recall, right after the pandemic was announced by WHO, just for a few days after that, the genome sequencing of the Wuhan virus was made available globally. So, in short, the exploratory phase of the COVID-19 vaccines, instead of going up to 4 years, it was cut short to 8 months to 12 months. Now, next stage is the preclinical. So this is the stage where now you have your vaccine candidate and you wanted to test it in the laboratory, of course, with animals, where we use mice, or monkeys. This is where you test the safety of your vaccine and check into toxicology studies. You test the purity and stability of your candidate vaccines. Typically, these last for 1 to 2 years. Now, I'd like to correlate again to the COVID situation that we are in now. The preclinical stage has been shortened with the COVID-19 vaccines. In the standard way, this usually takes, let's say, 2 years. But because there was this agreement with governments globally that once a vaccine is produced, they will be purchasing the vaccine. So instead of going slow with testing in the laboratories, this was expedited. And so what we saved here is time. Now, during this phase, there are 2 documents that is important for the vaccine manufacturer to prepare. One of them is the target product profile, because if your preclinical studies are successful, you should now have an idea what dose to use when you have your vaccine in development, the dosage forms, what are the contraindications, would you want some warnings or precautions associated with your vaccines, what were the adverse events seen during the preclinical stage, and how do you want your vaccine to be used, what is your target population. So this is called the TPP. Then the other document would be your CDP, which is the Clinical Development Plan. Now, this is a very complex document that tells the entire clinical research strategy of your candidate vaccine. So this is based on the TPP that you have prepared. Now, listed here are the 6 main elements that CDP should include. Then once your TPP and CDP are ready, you are now ready to start your clinical development. And if we say clinical development, there are 3 phases standardly, phase 1, phase 2, and phase 3. Now, for phase 1 study, this is what we call the first in human. This is the first time that your vaccine will be tested in humans. And conducting phase 1 study, usually we start with adults. We do not start with pediatric population. And the primary objective is, of course, the safety of your candidate vaccine. And secondary, the immune response, whether your vaccine would really elicit immune response to your target population. After the phase 1, you get into phase 2. Of course, even in phase 3, you always have safety as a primary or a secondary objective. as well as immunogenicity. In phase 2, what are the activities that you can perform? You can do your dose selection, your age de-escalation studies, your lot-to-lot consistency can be done. And most of the time, there could already be a guess on some form of correlates of protection during the phase 2. Now, in phase 1, you include around 10 to 100 subjects. In phase 2, you can include hundreds to a few thousands subjects. Now, if your phase 2 is successful, then you can proceed to your phase 3. Now, phase 3 is where you conduct a larger study involving sometimes as many as 40,000 subjects, where you really look into the immune response of your candidate vaccine to the targeted population. Now, of course, the golden standard in conducting usually phase 3, it would be a double-blind randomized studies. Now, if your candidate vaccine is the only vaccine available in the market and there's no comparator that can be considered, you are allowed to perform the study using a placebo. However, if there are comparators in the market, it is advised that you do a non-inferiority study. Now, just for an update with the current COVID pandemic, yes, the vaccines now, COVID vaccines now, they were initially conducted using placebo because there were no COVID vaccines available. But now that a number of vaccines are under EUA, the second wave of COVID vaccines under study now are really advised to use a comparator vaccine. That is the current vaccines that are having EUA. Now, also, I wanted to share with you a paradigm shift in the conduct of clinical studies. As I said earlier, usually the standard is 10 to 15 years. Now, there is this adaptive seamless design that is being adapted in the pharmaceutical industry, where you combine your phase 2 and phase 3. Now, in combining this, you save a lot of money, you save a lot of time, as well as minimizing the number of participants in the study. This is one example of a study that we are currently conducting. It's showing you that phase 2 and phase 3 had been combined, and this is all in one protocol. So that adaptive seamless design is a reality during this time. Now, if we go to the next step, which is the regulatory approval, it is important for the vaccine manufacturer to know that as soon as your phase 3 studies are successful, you can already prepare your dossier. The dossier are the documents that you have accumulated right from the exploratory phase, preclinical, clinical. You combine all these documents and submit them, and we call them the dossier. Now, showing here to you are the different regulatory agencies globally. So it is not only FDA, which is for US. It is not only EMA, which is for Europe, but other countries have, just like UK, you have MHRA, Japan, you have JPMA, and although the requirements of each regulatory authority is uniform, they have their specific requirement depending on the local need. Now, this is to show you that consultation with regulatory authorities are really advised, right from the very early phase of your study to the continuing phases of phase 1 and 2, even after your phase 3 studies prior to launching your vaccines. So consultation with regulatory agencies would guide the vaccine manufacturer on how to go forward with its development. Now, in order to have licensure, these are the 4 things that a vaccine manufacturer has to consider. First of all, your CDP really has to be robust and comprehensive. And if there are gaps during clinical development, ensure that all these gaps have been addressed. And then you have to prepare your risk management plan. And by the way, this is a requirement under ICH GCP. So all throughout the clinical development phase, the risk, how you handle the risk should be clearly defined. And once you submit your dossier, ensure that it is in the correct format according to the country where you are submitting your dossier. Now, I mentioned dossier, so this is equivalent to the common technical document. And this triangle, as soon as you see this triangle, you already know that you are referring to the dossier. So it comprises 5 modules. And if you look at the center of the triangle that's colored yellow, this is the bulk of the clinical development where you accumulate all the things that you have done right from the exploratory phase until you completed your clinical development. Clinical study reports are also compiled, as well as non-clinical study reports, which refer to those experiments, tests that you have done during your preclinical development phase. Now, then the next stage would be manufacturing. I just wanted to emphasize that even if you get licensure, registration, marketing authorization in the regulatory and approval phase, life cycle of the vaccine, the management of the life cycle of the vaccine does not end there. So constantly you will get facility testing, and constantly you would have a test on vaccine samples, because regulatory authorities are also monitoring this, the way the vaccines are being manufactured, to ensure that they are safe for humans. Now, after that, the last stage would be quality control. Quality control, so it's a continuing monitoring of manufacturing. Under quality control, this is where you monitor the adverse events of a product, of a vaccine. Now, when a vaccine is already in the market, we are still able to monitor the adverse events. This is reported as spontaneous reports. And for all those people who have used this specific vaccine and who had adverse reactions from the vaccines, these are constantly monitored. Now, I'd like just to emphasize that the World Health Organization is really very much engaged and very much supportive of vaccine development. Now, they provide recommendations as well as policies for all the members. And the most prominent one would be the SAGE recommendation, which would be equivalent to WHO global policy. And all vaccine manufacturers are really consulting WHO in their clinical development phase. Now, let's talk about vaccine supply. So once a vaccine is licensed, what are the ways to get this distributed to the people who need it? There are 2 ways, direct procurement and through the UN. So in direct procurement, that is, if you submit to specific countries, to their local authorities, and that specific country granted you a marketing authorization, meaning your vaccine is licensed in their country, and so they can purchase your vaccine and you can deliver to that specific country. And the other one, through UN procurement, first you have to be included in WHO PQ. PQ is the pre-qualification, or you would be in the emergency use listing. Now, just to inform everyone, before you can get an EUL, you have to be in the priority listing. And currently, as of last month, we have checked with some WHO collaborators. The only vaccine that is under priority listing in WHO PQ is the COVID vaccine. So for now, it is only COVID-19 vaccine that is allowed to undergo an emergency use listing. So these are the requirements of how to submit for a WHO PQ. As I said, you have to be in the priority list. And then you should have been approved or licensed in the country where the vaccine is manufactured. And your responsible NRA should provide oversight to the use of your vaccine. Now, in the current pandemic that we are in, as early as December 2020, they formed a technical advisory group for the EUL. So this was only formed because of the pandemic. Now, as you can see, you have to meet the 3 criteria for you to be eligible to apply for an EUL. The first criteria, that your vaccine candidate is intended to address a disease that is life-threatening and that may cause a pandemic. In short, COVID-19 vaccines fulfill this criteria. Next, the existing products have not been successful in eradicating disease. And the COVID-19 vaccines fulfill this criteria because we are still in the pandemic. And even if we have the EUL, these are just, helping us not to suffer in death or severe hospitalization. And third, the product has to be manufactured under good manufacturing process. So there, I hope I gave you a brief description of what clinical development is. Thank you very much.
Thank you, Zenaida. That was a very nice opening lecture for today's presentation or today's agenda. And I can see on the group there are a lot of questions being raised. And since, unfortunately, Zenaida could not join us for this live Q&A, I'm going to project 2 of the questions that have been generally asked in the Q&A, and we'll try to answer those. So the first question, which I saw in the chat and the Q&A box quite frequently, is it takes many years to develop new vaccine, a decade or so. And how did we manage to get it within a year for COVID-19 vaccine development? And, if you ask people who are like anti-vaxxers, they also see some sort of conspiracy in that, right? But I think there is pure science behind that and how we managed. So this is the answer. I don't know whether you can see this, especially the diagram, if you take a look at that on the right-hand side. So the upper part of the diagram talks about the normal process, the traditional way of development of various vaccines. So it goes from the design and exploratory preclinical studies, which generally takes years. Then we have the process development part, preclinical studies, which varies anywhere between 2 to 4 years. Then you go for the investigational new drugs submission, which is IND. Then there are sequential clinical trials, phase 1, phase 2, phase 3. After that, we get the licensure or the BLA, as we call it in vaccine terms. And then it goes for the regulatory approval for the global use of the vaccine. And then there is large-scale production of the vaccine to be available in the market for the people who need the vaccine. What has really helped us for the COVID-19 vaccine is, since there was pre-existing knowledge about coronaviruses from the SARS outbreak and the MERS outbreak that happened before, there were a lot of manufacturers who were having this knowledge, and there were a lot of technologies which were developed for these vaccines. So we started to work on it quite early on. And then also we tried to do the parallel pre-existing preclinical studies so that we can go to the clinical trials quite quickly. And then we ran multiple parallel clinical development programs for various of these vaccines so that we can compress the timeline without compromising the safety or efficacy which is required, the scrutiny of those vaccines. And one of the things that has really helped us having all these global players come together is many of the manufacturers, they started manufacturing the product even during the clinical development when the product was not licensed at risk. So that when the product is licensed, and if we see that this is suitable to be used for the mass production, they are already ready and they can start supplying very quickly. So with that, we basically were successful in compressing the timeline from, let's say, 10 to 15 years, which is the traditional way, up to 1 to 2 years maximum that we have seen for COVID-19. And you can see on the text on the left-hand side, there are several COVID-19 vaccines which are now authorized for emergency use in multiple countries. So this is a great achievement of the science. There is no conspiracy. There is nothing that we have cut corners. So we should all feel quite satisfied with the way the science has progressed so that we can actually have response globally for a particular disease which can be deployed within a year of its occurrence. The second question, I think, which is quite commonly asked in the Q&A part is, what are the requirements for clinical data in the phase 3 COVID efficacy trial? So if you refer to the US FDA guideline, there are multiple requirements there. The first requirement is the point estimate for the efficacy should be at least 50% with at least 30% on the lower bound. You need to have the safety data accumulated from phase 1 and 2, which will help us for the phase 3 data also, so that the safety requirement or the safety assessment that we need to do for the candidate is taken care from all the way phase 1 to phase 3. Then one of the requirements is for the phase 3 samples or phase 3 participants to be followed at least for 2 months post last dose of the primary series. Generally, COVID vaccines are given as 2 doses, a few weeks apart. So we have to make sure that they are followed up at least 2 months after the last dose in the primary series. And then there is certain requirement of number of participants or vaccine recipients who should be exposed to that vaccine at least 1 month after the primary series, which in this case is 3,000. And there should be sufficient cases that we need to have of COVID-19 among the study subject to support the low risk of the vaccine-induced respiratory disease or the COVID-19 related severe illnesses. So this is the requirement. I can only take 2 questions at this point in time since we don't have Zenaida. So we can move to the next presentation that we have for today's agenda. So the second speaker that we have now is again somebody that has worked in IVI for a long time, Dr. Thong-Yong Kim. She is our head of biostats and data management, and she is going to talk on the statistical methods for vaccine development. Dr. Kim.
Good morning, everyone. I'm Thong-Yong Kim, working in IVI as a head of biostatistics and data management department. I've been working in IVI since 2000. For the last 20 years, I've been involved in every phase of clinical trials, as well as several observational studies of mainly cholera and typhoid disease and vaccine trials conducted in Asia and African countries. And recently involved in chikungunya vaccine trial and the COVID-19 vaccine trial to support statistics and data management. It is my pleasure to have a talk in the 20th Vaccinology course at the webinar. I'm going to talk about statistical approaches in vaccine development and share my experiences in the statistics with you. I hope this opportunity can help you to extend your knowledge and apply it to your research. Before moving to statistics, we'll briefly see the overview of vaccine development and the approval phases. And as a statistician's point of view, I will talk about what are study objectives in the vaccine trial and what are corresponding study endpoints and trial design characteristics. When it comes to statistics, we'll see the three major statistical tests for clinical research evaluation and the statistical method based on discrete type and continuous data type. Finally, I will bring you up COVID vaccine efficacy trial design and the points, primary analysis, success criteria, and sample size calculation in the trial design, and references and Q&A. Once complete the preclinical stage, the investigational new drug will be submitted to the regulatory authority for their approval to conduct the clinical trial. So all the clinical trials which are planning to conduct in the country should be approved by the national regulatory authority before starting the study, as well as approval from the ethical committees. After completion of phase 3 efficacy trial, new drug application is submitted to regulatory. For approval, the company also must show their large-scale commercial manufacture and produce vaccines of the required quality. After the vaccine is launched in the market, safety monitoring will be conducted for collection of safety data from a more vaccinated population. In each clinical trial phase during vaccine development procedures, statistical considerations are critical to make sure if the design is adequate, appropriate data has been collected, the collected data has been properly summarized, and inference and interpretations are appropriate for the study objectives. So please keep in your mind statisticians' involvement from the early stage to the end of the study. In the clinical trials, the study objective is a big picture of the experiment done in the clinical research. Why we do this experiment? The answer is to establish the effect of the intervention in the vaccine clinical trial. The study objectives will be to assess the safety and demonstrate immunogenicity and efficacy. In case of a formulation change, to demonstrate the equivalence of a formulation will be an objective. In case of a change of administration route, such as intradermally or intramuscularly injectable vaccine to microneedle patch to relieve pain in the injection site, the participants' satisfactions also will be an objective. There are specific objectives in each vaccine trial. Sometimes immune lateral consistency and then and immune non-interference with EPI vaccine will be considered a study objective in phase 3 study. To achieve study objective, what is the measured? This is a study endpoint. Depending on the objective, the measurement of endpoints is different. For example, for the efficacy trial, instance of a disease will be a study endpoint. If there is one primary objective, there is a corresponding primary endpoint. Sometimes it could be one or more. The primary objective and endpoints need to be specific because the primary endpoints are typically measured that address the main research questions. We also have secondary objective and tertiary and exploratory endpoints. These endpoints, which may generally not be sufficient to influence decision-making alone, but it may support the claim of a primary objective by demonstrating additional effects. Each endpoint needs statistics to summarize data or show the statistical inference. Please keep in your mind, there will be the increasing number of statistical tests as increasing number of endpoints. Here is the list of endpoints mainly considered in the vaccine trials: safety, immunogenicity, efficacy, effectiveness, and lateral consistency. We may consider several trial design characteristics to design vaccine trial. In a comparative trials, superiority versus non-inferiority trial. And depending on the study design, we need a placebo control versus active control. And the study needs to be randomized control versus open label due to ethical and safety issues. And cluster randomization needed to avoid the contamination of the responses versus individual randomization. Sometimes age stratification is necessary due to varying disease instance and or the immune response by age. Method of level of blinding and the time of study level unblinding is also need to be considered. The dosing regimen, treatment duration, measurement time points, and study follow-up duration also need to be considered. Sometimes the study may need that interim analysis for moving forward to next phase after dose selection or to initiate the licensure process. The trial design is really depending on the phase of development and characteristics of vaccine or disease. Here is a few characteristics under each phase 1, 2, and 3. You can see more details in these slides. From now on, in the following few slides, I will talk about 3 major statistical tests. The first one is the superiority test. Most of the clinical trials are usually performed that the new vaccine is superior to the control vaccine. The objective will be to determine whether test vaccine is effective compared to control. From the statistical inference, the study objective will be demonstrated by use of p-values. Usually the threshold will be used 5% or 2.5%. Here is the superiority hypothesis here. The alternative hypothesis is to demonstrate the test vaccine is better than compared vaccine by statistical significance threshold. Here we have 7 examples, 7 studies. Out of 7, study C and F demonstrate the new vaccine is effective than control. Even study E showed a statistically significant, but it demonstrated the control vaccine is effective. All the other studies didn't show the new vaccine is effective. Next, we may consider non-inferiority study design if clinical trial is designed to have a high chance to demonstrate a clinically at the relevant difference. The clinically relevant difference is defined as a smallest clinical meaningful difference between treatments. We call the clinical meaningful difference as a non-inferiority margin. It must be predefined and in the protocol. So from the statistical inference, it will be demonstrated by use of a confidence interval. Let's think about why the non-inferiority test design is necessary. When compared to existing vaccine, the test vaccine may be expected not to be better on the primary endpoint, but it may be better on secondary endpoints. Or it is safer, it is cheaper to produce, or it is easier to administer. So, for example, in the vaccine development, the same pathway of a vaccine can be approved based on the immunogenicity comparison with a marketed vaccine without efficacy trial. So please keep in your mind, non-inferiority cannot be demonstrated with non-significant p-values from the superiority test. Here is the non-inferiority hypothesis. So the alternative hypothesis of non-inferiority test is to demonstrate the test vaccine is non-inferior to active control if the lower bound of the confidence interval is greater than the clinical non-inferiority margin. here is I also brought 7 studies. So study A, B, C, E, F demonstrate the new vaccine is non-inferior to active control vaccines. Even study C and F showed a statistical superior to active control. The study E showed non-inferior to active control, but statistically inferior to active control. All the other studies didn't show the new vaccine is non-inferior to active control. Regarding non-inferiority margin, this is the ICH guidelines. The margin should be a statistical reasoning and then clinical judgment. Determining non-inferiority margin is judged by the clinician and must be predefined in the protocol. So in the vaccine trial, for the WHO pre-qualification, WHO required to use -10% for zero conversion difference and 0.67 for GMT ratio. Sometimes 0.5 can be acceptable for special cases. Now we move on equivalence test. In case of formulation change, the study may want to demonstrate the multi-dose formulation is equivalent to the single-dose formulation. For clinical equivalence, there needs to be a strong clinical rationale for the accepted margin. In a vaccine trial, for lateral consistency, the equivalence margin for GMT ratio will be 0.67 to 1.5. The equivalence margin for zero conversion difference will be at the plus minus 10%. This is the equivalence hypothesis. The alternative hypothesis to demonstrate the test vaccine is equivalent to active control if lower and upper bound of the confidence interval is within clinical margin. So out of 7 studies, study A and E and F demonstrate the new vaccine is equivalent to the active control. All the other studies didn't show the equivalence. This is a summary of interpretation of superiority, non-inferiority, and equivalence test. Statistical superiority will be demonstrated if the lower margin is lower bound is greater than 0. And the clinical non-inferiority, if the lower bound of the confidence interval is greater than the lower margin. In this sense, we call the study demonstrated clinically non-inferior. And clinical equivalence, if the both bound is within the lower and upper margin, we call the study demonstrated clinically equivalent. And sometimes the lower bound of the confidence interval is greater than the upper margin. We call the study is clinically superior. Now we move on statistical method. The table shows 2 different outcome variable types. One is a nominal scale, like yes or no, and the other one is a continuous scale, like antibody titers. The instance of a disease, zero conversion, and safety events will be measured at the nominal scale. The immunogenicity endpoints like antibody titers will be measured as a continuous scale. The nominal scaled endpoints will be summarized as a percent, rate difference, odds ratio, relative ratio, and this will be tested using asymptotic G-test and logistic regression model. Here is an example how the nominal data will be summarized. You can refer in details. And continuous outcomes. The antibody titer is usually observing at the reactions at the concentration which are multiples of each other, like 2 times, 4 times, etc. It means the log scale is concentration increasing arithmetically. So geometric mean and geometric standard deviation is better to summarize the data. The two groups comparison will be tested using a two-sample t-test or a g-test. And if we have more than 2 groups, the analysis of a variance model will be used. From now on, for a few slides, I brought the COVID-19 vaccine efficacy trial design. This is an example of COVID-19 efficacy trial design. You can look at in details. Next one is example of study endpoints for COVID-19 vaccine efficacy design. You find the primary and secondary endpoints. Under SARS-CoV-2 infection, COVID, asymptomatic COVID and asymptomatic COVID infection. So all this will be considered as efficacy endpoints. The primary analysis is the evaluation of vaccine efficacy. Vaccine efficacy is calculate 1 minus the hazard ratio if the study use a proportional hazard model. So next slide is about sample size calculation under the following assumptions given target vaccine efficacy and background incidence rate in placebo group, as well as right side, the graphs shows various sample size based on the varying vaccine efficacy assumptions. In summary, I'd like to summarize a few points of statistical perspectives in the vaccine development. We learned the vaccine study objective endpoints and design are determined by depending on the phase of development and development stage of a vaccine and characteristic of vaccines. The superiority, non-inferiority, and equivalence tests are typically used in the vaccine development. We needed to consider the study design, hypothesis, and data type when we choose a statistical analysis model, as well as it is necessary to check underlying assumptions of the statistical model. Lastly, let's report not only p-value, but also the estimate of the effect and the confidence interval. I start my presentation now. Here are references that I referred for my presentation. Thank you for your attention.
It's never easy to explain the biostats and the complexities that we have in statistical analysis, even in one semester. But thank you so much, Dr. Tong-Yong Kim. You have explained that in less than 30 minutes. So again, quite happy with the way the presentation is going and the questions are coming. So now, since Dr. Kim has joined us for the live Q&A, I'm going to pick up some questions from the audiences. So the first question, Dr. Kim, for you is, what is the role of statistical analysis in non-clinical studies? Can these statistical methods be adopted for preclinical vaccine development, in particular in the conduct of animal studies?
Thank you for these very nice questions. Usually in clinical, non-clinical and preclinical trials, so there are not many subjects, the mice or the rats, a very small number of subjects is tested in the preclinical trials. So usually those small number is not meet the statistical power. So however, we may compare the groups. So in that sense, the stats analysis is also necessary to provide us some kind of the p-values.
Thank you so much. So then we have a longer question. I will try to summarize that. The question is, with respect to comparative non-inferiority studies on COVID-19 vaccine, what are the specific consideration in terms of study outcome, statistical analysis, especially on the desired margin between immune responses of the test vaccine versus existing? Is it still 10%? Or is the margin different for COVID-19 vaccine?
Yeah, basically at the non-inferiority study, we even if in COVID study and other study as well, so we are considering the geometric mean titers and as well as the zero conversion rate or zero positivity rate can be also considered from the study outcomes. So in terms of the margin, so it's always difficult to define if whether this margin is clinically or statistically reasonable. So basically, at the WHO pre-qualification requirements, they mention about the non-impaired margin minus 10%, and also when we use the geometric mean titers in that sense, 0.67. I think still those margin is acceptable for the COVID-19 study as well.
Okay, thank you so much. The next question is, if in a study there are multiple interim analysis planned, how should we control for multiple comparison?
Yeah, so in many studies, research scientists wants to compare the many ways. However, we need to really focusing on what is our primary comparison. So for that, we need to define our alpha as well as in the several interim analysis, we need to think about the multiple adjustment. For example, you may consider the bone perennial method is a very conservative method, and also you can think about the other ways as well. So number of tests is increased in the sense that you need to also lose a lot of you need to spend a lot of times of alpha. So you need to, when you design the study, when you define the primary objectives, when you define the primary endpoints, you need to also consider how many tests is necessary to meet the study-wise alpha.
Perfect. So now the last question. If the study is planned for non-inferiority, can you still do the superiority testing at the end of the study if the study was initially planned for non-inferiority?
So basically, before when we use the non-inferiority study, means that we already assume that this vaccine is already superior to the placebo controls. So I think we don't need the necessary placebo groups in the non-inferiority study. However, the ethical issues, some regulatory or EC body may request to you.
Perfect. So we actually had time for these 4 questions. Thank you so much, Dr. Kim. And then now we can move to the next presentation in the afternoon. So we have now Dr. Flor Munoz, who is joining us from Baylor University. Flor is actually giving lectures in IVI Vaccinology course for quite some time, and she is going to talk about maternal immunization. Professor Munoz.
Hello, everyone. My name is Flor Munoz. I'm a pediatric infectious diseases specialist in Houston, Texas, and I work at Baylor College of Medicine and Texas Children's Hospital. I'd like to go ahead and share my screen today and present to you the topic of maternal immunization. These are some of my disclosures for your information. And the objectives of today is to discuss the concept of maternal immunization, talk about different pathogens and the research in pregnancy and women of reproductive age, as well as focusing on SARS-CoV-2 in pregnancy. To set the stage, it's important to take into consideration the fact that pediatric mortality globally remains a problem. Although we have seen a decline over the last 3 decades, substantial decline as a matter of fact, we see that mortality in children under 5, and especially in neonates, remains a significant burden globally, especially in countries of lower resources, as you can see depicted in those maps. Over 2.5 million deaths continue to occur in the world, and you see that these represent about almost 50% of the mortality under 5. Maternal mortality has a similar distribution, and it has also declined over time. Nevertheless, you see that as of most recent data, there are still about 830 maternal deaths occurring every day in the world. It is important to keep in mind that maternal immunization could address this problem of maternal and neonatal mortality. So the rationale of maternal immunization is really that we know that children are at high risk of morbidity and mortality from infection, usually in the newborn period more than any other time in life, and that they do depend on maternal antibody to resist infections in early life. We know that specific antibody can protect mothers and their babies against some infectious diseases that we have already evidenced through epidemiologic studies, including tetanus, herpes streptococcus, influenza, and RSV. And we also know that in some cases, moms have low levels of antibody, for example, in pertussis. And those antibody levels can be enhanced or optimized during pregnancy through maternal immunization. So this is really a concept of boosting levels of antibody in mothers so that they can transmit that protection to the babies during a period of increased vulnerability and protect them. This is a simple concept in terms of transplacental antibody. We know that the placenta is an organ that has a selective and active process of transporting antibodies from the mom to the baby through a receptor, that is Fc receptor, and it's specific for IgG. You see that in the graph on your right, you see that there is an exponential increase in the transfer of antibodies that occurs starting at about 17 weeks of gestation to the end of pregnancy, so that at the end of a normal 40-week gestation, you have more antibody on the baby's side. We know that the half-life of this antibody is about 30 to 40 days, and that the higher the antibody level, the better the protection will be and the longer the duration of protection. But there are many factors that can affect this process. For example, the level of maternal IgG, the type of antibody, if it's the different subtypes of IgG. We know that there's different receptors, and again, specific composition of these antibodies, such as glycosylation, may affect the process. But the interval from vaccination to delivery, the gestational age at the time of vaccination, and other maternal and placental factors or conditions could also affect the transfer of antibody. So when should a woman who is pregnant be vaccinated? I think that it's important to note this concept because it is something that is in concurrence with different organizations such as the WHO, the CDC, the CIP, and even obstetric groups. So we know that if women have a high risk of exposure to a particular disease, and that infection or that disease is a special risk to the mother or the fetus, and you have a vaccine available that is safe and unlikely to cause any harm, you see that the risk of the disease is much greater than the risk of the vaccine, and therefore it is justifiable to offer that vaccination to the mother. You can see that the statement of the WHO on vaccines and pregnancy matches this concept, so that a woman who is pregnant should not be deprived of receiving a vaccine to protect herself or her baby if there is, again, a high risk to her or the baby and you have a vaccine that works. You see in this table the generally recommended vaccines, which are usually tetanus. As of this year, TD is the one that should be favored for paternal immunization. Influenza vaccines, inactivated vaccines, and in some parts of the world, a serial pertussis vaccine should also be used in the form of Tdap, or tetanus, diphtheria, and serial pertussis. You see that to the far right, there are some vaccines that are contraindicated because of a theoretical risk of potential infection to the fetus. Those are the live vaccines, so they're usually avoided during pregnancy. But the vaccines in the center can be recommended in special circumstances, and including there is SARS-CoV-2 vaccine, where you have an outbreak, you have an epidemic or a pandemic, and you have the option to protect the women if they meet those criteria, where the disease is much worse than the vaccine, potential risks, and you have the opportunity to protect both moms and babies. So we know that this concept is applied widely in various circumstances. We need to know that vaccines as of today have not ever been licensed specifically for pregnant women. There are actually no vaccines that are licensed only for pregnant women. But the vaccines that are already licensed for other populations, such as Tdap and the influenza vaccine, can be utilized. And this is an example of the regulatory agency, the FDA, that says in their labeling that these vaccines are not contraindicated for pregnant women and that they can be used based on data that is available in multiple studies. But that safety, of course, needs to be monitored. This is an important concept. So I have a very busy slide here, but I wanted to share with you the fact that maternal immunization is not a new concept at all. It has been present since the beginning of the vaccines. We have to the far left there the vaccinia or smallpox vaccines. and all the way to more recently, vaccines against influenza, pertussis, and we will see in a moment, even COVID vaccines. And you see that over time, there have been different infectious diseases that affect the mother and or the baby, such as tetanus, smallpox, pertussis, streptococcus, and even RSV, influenza, that are the targets of this research that has been taking place now over decades. And so it's important to keep that in mind, that this is nothing new at all. We have to continue with this work. However, there are some key issues that you need to keep in mind. For example, as we mentioned, no vaccine is specifically licensed for pregnant women yet, because the vaccines are usually recommended for pregnant women on the basis of risk benefit. And we yet have to understand better the potential impact as a public health strategy that maternal immunization has. We have seen it in cases of outbreaks of influenza, Ebola, and more recently with COVID-19, that inclusion of pregnant women in the immunization strategies can be an important public health strategy. Furthermore, there are new vaccines that are being developed for pregnant women, and those include, for example, RSV, group B strep, and even CMV vaccine, cytomegalovirus. And you can see that as a research subject, pregnant women do need to have the potential benefit of being part of research, because we need to assess the safety of the vaccines on them. We need to be able to determine the safety and also the efficacy in them and also in the babies. And we need to understand any knowledge gaps that remain in place. So there is a lot of work regarding the ethics and the regulatory requirements for vaccines in pregnant women. I'm going to cite some examples briefly regarding maternal immunization. So maternal tetanus immunization is one of the mainstays throughout the world. You see that this is a program that started in the 1960s. 2 doses of tetanus vaccine are able to reduce by 96% maternal neonatal tetanus mortality. And you see that even with an imperfect coverage, because it's not 100%, you have a significant decrease in impact in this disease. So this is a platform that is key and it works. In terms of influenza immunization during pregnancy, you can see here some of the milestones. We recommend vaccination for women every influenza season. It is actually an essential element of prenatal care in the United States. And you see the progression from a recommendation in women at risk in the 1960s to now vaccinating all pregnant women at any trimester in gestation. And after the flu pandemic in 2009, there was certainly now a global goal to vaccinate pregnant women as a priority against influenza, so that we have to include that in many countries that already have influenza programs in place. In addition to that, I have to tell you there's data after many, many years of using the influenza vaccine in risk-benefit situations. We now have data from studies that occurred in the early 2000s. This one is one of the key studies that occurred in Bangladesh, where you see that women who were vaccinated either with the flu vaccine or pneumococcal vaccine during the 3rd trimester were able to show a decrease in respiratory illness by 36% in the vaccinated mothers compared to the control group. And in infants, you see a 63% reduction in laboratory confirmed influenza in babies of vaccinated mothers, and also almost a 30% reduction in respiratory illness. So this was one of the first studies to demonstrate efficacy or effectiveness of the vaccine in both mothers or babies. And from there were 3 additional studies, one in Nepal, one in Mali, and one in South Africa, that were able to demonstrate that with different controls, a vaccine efficacy of the flu vaccine given during pregnancy of about 42% in decreasing PCR confirmed influenza, 60% if given after pregnancy, and then during the full period, you see of observation about 50%. So it works in pregnant women, and it also works in babies. You see here again a similar range of efficacy in the babies against influenza confirmed by laboratory, even if the babies were not vaccinated, but the mothers were vaccinated. So we can tell moms that this vaccine protects them, and it also protects the babies. As a matter of fact, because of these studies, influenza vaccine is one of the first in this area where you do have in their package insert, in their label, an instruction to indicate that it is for prevention of disease in pregnant women and also in the infants in the first 6 months of life. And this is approved in Europe and also in Australia. Now, you see here other studies more in real world situations where It has been demonstrated through registries and through other case control studies that the efficacy of the vaccine against either laboratory confirmed influenza continues to be in the 50 to 60%. But more importantly, the PREVENT study at the bottom, which looked at several countries, at least 4 countries, a 40% protection against hospitalization from influenza in pregnancy. So it is an important point that we are seeing that you can still have influenza infection, but you will protect moms against the most severe consequences that result in hospitalization. Another disease that we have a maternal immunization program for is pertussis. And similarly, the goal is to protect the babies primarily because of pertussis disease being a significant problem in the first 2 to 3 months of life, where you had an increased mortality over time. And we started vaccinating mothers in 2012, both in the US, in the UK, and other countries. And you can see here the follow-up of those studies showing a very significant effectiveness in preventing infant pertussis of about 69 to 91%, greater than 90% prevention of hospitalization, and about 95% prevention of death in the babies of mothers who were vaccinated. And this has resulted in a significant impact where you almost don't see any mortality from pertussis anymore if women receive the vaccine during pregnancy. So highly efficacious. One of the potential drawbacks we have seen is in some studies looking at infant immunization, primary immunization. If babies receive the vaccine, if babies were born to mothers, that received the vaccine during pregnancy, there could be a blunting or maybe interference in their immune response to their primary active vaccination. But this has not resulted in any clinical changes. And so it is a trade-off that you will see some decrease in the actual antibody titers that they get. But this maternal immunization does not interfere with priming or with the ability of these babies to boost their immune responses through routine primary infant immunization. And here we have to consider as well other diseases. So it used to be a foreign topic, talking about giving vaccines to pregnant women, but we have seen a number of outbreaks such as Zika that have really made us think very, very carefully about maternal immunization as a strategy to protect mothers and babies. And in this case, because it caused congenital infection, and certainly it's a disease that has come and gone, but it's possible it will come back. And we should be prepared to prevent it either through vaccination in women of childbearing age or even during pregnancy in the future. Another one is Ebola, where in this case you have a disease that affects persons of all ages, and including pregnant women significantly. And initially, pregnant women were not included in the campaigns of vaccinating people during the different outbreaks in Western Africa. And you see in 2019, eventually with this risk-benefit assessment, it was determined that pregnant women should be included in these vaccination efforts because of the higher morbidity and mortality of Ebola in this population, moms and babies. So let me spend a few minutes now talking about COVID-19, because we also have learned a lot about this disease. And you can see here some of the early data from the United States where we started to note that women who were pregnant compared to those who were not pregnant in the first months of the pandemic really had an increased risk of hospitalization, about 5 times greater ICU admission, and the need for mechanical ventilation in the United States, maybe even death. And the data from the right is from the Living Systematic Review, more global, where you can see that it confirms that indeed pregnant women are increased risk for these complications, and even the babies can have increased risk for preterm birth or being in the hospital. So clearly, it met that criteria of having a disease that could increase the risk for moms and the babies. Other studies, just to point out briefly, you can review on your own. This is just another inter-COVID study where you can see that looking at different parameters, having diagnosis of COVID in the mothers and or the babies shortly at the time of delivery would increase the risk for neonatal morbidity, perinatal morbidity, and even mortality with different indices that were evaluated in this particular study. So 4 or even up to 6 times increased risk for different outcomes depending on the positivity. And then more recently, we have been dealing with the Delta variant. And yes, indeed, the Delta variant is more virulent, it's more severe, and even in pregnant women as well. So this is a study, an observational study from the UK, where they looked at a composite indicator of disease and looking at different periods from the time when they had wild type COVID-19 or SARS-CoV-2 to the Alpha variant, it's known also as the UK variant, and more recently the Delta variant. And you see that the composite indicator of severity continues to increase over time, indicating that the Delta variant is worse in terms of morbidity compared to Alpha and compared to the wild type. And the morbidity that you see in these periods of time is that there is significantly increased risk for pneumonia, for respiratory support, and for admission to the intensive care, as we have seen before with the Delta variant compared to the other variants. Importantly, no fully vaccinated pregnant women were admitted during this period of time. So the vaccine is working, but if not vaccinated, women will be at risk. And then just to point out, WHO has other studies ongoing, the Unity studies that are looking at the impact of COVID-19. This data is critical to assess the burden of disease in pregnancy and in the newborn so that we can implement interventions for prevention, such as vaccination. So what about vaccine and what about safety? You know that in the United States, we began vaccinating pregnant women at the same time as the general population in December of 2020. There is an established registry where women can register there and be able to assess the outcomes of pregnancy. And it's impressive, as you see from more recent data, nearly 150,000 women have been included in this registry, of whom about 5,000 are in this pregnancy detailed study report. The most recent publication coming from this registry is here in the New England Journal, where you see that the two vaccines that are given in the United States, Pfizer and Moderna, mRNA vaccines, really result in no more reactogenicity in pregnancy compared to non-pregnant people. Actually, all of these indicators, you see the dark blue bars are pregnant women, especially fever, for example, is less commonly seen compared to non-pregnant women. So it's very important to know that the vaccine is safe. In addition to that, when you look at outcomes of pregnancy, there is no increase in some outcomes, such as pregnancy loss of different types, spontaneous abortion or stillbirth, preterm births, or other anomalies or neonatal death. It's not higher than the background, which is quite reassuring. And this is the type of data that allows us to continue to use the vaccine in pregnancy. As a matter of fact, you might have seen that ACOG most recently in July of this year has changed from a guidance of favoring vaccination in pregnancy to actually recommending that all pregnant women should be vaccinated against COVID, because we know that the vaccine is safe. We know that there are many more people becoming infected, and women are getting infected, in particular with the Delta variant, and the currently low vaccination rates need to be improved. So this is why we need to continue to vaccinate pregnant women as an important recommendation. And this, again, for you to look at your leisure later on, as of more recently, this is the progression of the guidance. Needless to say, WHO and others had initially recommended against vaccinating women in pregnancy. And gradually, as again, some of the data, especially from the US, came along, then from Israel, more safety information was available. And now, as a matter of fact, all these recommendations are harmonized, indicating that pregnant women and lactating women should be vaccinated during pregnancy. Keep in mind that it depends on the type of vaccine. The data that we have is mostly with mRNA vaccines at this time. And so just to point out, there are a couple of resources here for you. One is PROMIT. This is just showing at a website from the Johns Hopkins School of Medicine, and you see how you have different policies throughout the world regarding the use of vaccines in pregnancy for COVID-19. And they actually give you the utilization of the different types, whether there's a recommendation in pregnancy or lactation. And you can see lactation, for example, is just like non-pregnant groups where they can have any vaccine. But for pregnancy, it's a little bit more restricted. So it's a paradigm shift, just mainly to finish this point. Now we are including pregnant women in clinical studies. We're including pregnant women in situations such as the COVID-19 pandemic. And it's important to keep in mind that women are able to consent and they are able to participate. So we are now just not excluding, but we need to justify if we exclude them from any reason, from any research, because they should benefit from research. And then here is just a list of different activities that have occurred throughout the years. You see that there are many other groups and many other activities that have resulted in this paradigm shift from regulatory changes, and a lot of research that has been supported, and then global support through obstetric organizations, for example, in data such as the PREVENT guidance that indicates that especially in high-risk situations such as outbreaks, we do need to include pregnant women in research and in immunization implementation strategies. And you see that the sky is the limit. Here we have other work that is ongoing. For example, group B strep, it continues to be a problem in neonatal sepsis, neonatal meningitis, and the studies have been ongoing since the 1980s. The challenge here is to be able to match vaccines that are fully valent against the different group B strep strains that are adapted to the area, the region where they circulate, and be able to have safe and effective vaccines with good correlates of protection to be able to implement these vaccines. So work is ongoing, and it has the potential to decrease not just neonatal sepsis, but also stillbirth and also maternal morbidity, because GBS affects both moms and babies. And the other disease is RSV, respiratory syncytial virus, where you see it's mostly a disease of newborns. Several vaccines have been also evaluated. Fortunately, there are currently ongoing trials with RSV vaccines for maternal immunization in phase 2 or phase 3, looking at protection of the infant. More recently, the trial with the Novavax vaccine was able to give us information regarding the timing, optimal timing of vaccination, and the interval between vaccination and delivery. The longer the time, the better in terms of transplacental passage of antibody to the baby. And this study also demonstrated that if you vaccinate women against RSV, you could have in the infant protection not just against RSV, but all-cause pneumonia as well, compared to those who receive placebo. So potentially a huge impact. This vaccine, unfortunately, was not able to meet their primary outcomes, but again, other vaccines are in evaluation. And lastly, other diseases such as Lassa fever, which results in maternal death or fetal loss in about 80% of pregnant women who become infected, and hepatitis B, which is a lot more morbid and has a fatality in 30% of women who are pregnant who get this disease compared to 2% of the general population, should be targets for vaccines that would be given in the future to pregnant women. So these are my final thoughts. Again, maternal immunization works. We have seen the experience with tetanus, with pertussis, with influenza, and now even with COVID-19. And it can be an effective public health intervention to improve both maternal and neonatal outcomes. Vaccination with every pregnancy might be necessary depending on the goal, especially if the goal is to protect the newborn. And the optimal timing of vaccination might depend on the pathogen. So we need to understand maternal and infant immune responses. We know that protection for breastfeeding is a possible bonus. We didn't talk much about that, but that is very, very possible. And that we also need to consider the effect of maternal antibody on infant immune responses, such as the case of pertussis, when we look at these strategies, because we need to make sure that the infant immunization is not affected by maternal antibody. Lastly, we need to have good systems of safety surveillance and efficacy assessments once these vaccines are implemented. And implementation clearly is challenging. It's very important to raise awareness, education, have good resources, good support and leadership, as well as good communication strategies to make sure that these plans and these programs are successful. So with that, I will thank you for your attention today.
Thank you so much, Dr. Munoz. It was very great presentation on the maternal immunization part. Actually, we do apologize for the sound quality. The audio was probably not that clear. And then second point is Dr. Munoz could not join us live for the Q&A session, but I see a lot of questions being asked in the Q&A part or tab. So we'll try to get back to you with those answers from Dr. Munoz. So now we have to move to our fourth speaker of the on the agenda. We have IVI's Dr. John Louis Excler, and he's going to talk about post-marketing vaccine safety pharmacovigilance. Dr. John Louis?
Good morning, good afternoon, good evening. I am Jean-Louis Excler. I am the program director for new initiatives. I've been working at IVI over the past 5 years as head of clinical development and regulatory. And now for new initiatives as program director, I have 30 years of vaccinology experience, vaccine development, mostly HIV vaccines. Now, today I intend to discuss, to present the basics of pharmacovigilance. The goal of this presentation is to present you a post-marketing vaccine safety pharmacovigilance process. So we are in a unique era of COVID-19 pandemic that's triggered a lot of interest in vaccine safety and pharmacovigilance more than usual. The methodologies of vaccine efficacy and vaccine effectiveness assessments are well codified. However, the safety assessment is much more difficult. Usually it cannot be measured directly. It is inferred indirectly by relative absence of multiple adverse events following immunizations. And the study endpoints are multiple AEFIs. Only some are known, and some clinical syndromes with unknown cause that we call adverse events of special interest. Higher standards of safety are expected for vaccines. First, do not harm, prevent non-nocere. We have all learned that at the medical school. This is also a moral duty from a public health perspective. And the vaccinees generally are healthy versus ill for drugs, and vaccinations are universally recommended or mandated. So there is a de facto lower risk of tolerance for adverse events, and which leads to the search for rare reactions. Vaccines with less than 100 for 1 for 100,000 versus drugs for 1 to 4,000 events indicate the magnitude, the difference in magnitude of this risk tolerance. The studies of rare events are more costly and difficult and less likely to be definitive. Evolutionary success of immunizations requires adaptability. There is a need for an immunization safety system that is credible, effective, sustainable, appropriate, with scientific infrastructure, with governance, with transparency, no real or perceived conflict of interest, with a structure in terms of location, level, and stakeholders well defined, independence from pro and anti-vaccine stakeholders. It needs also resources in terms of staffing, funding, and tools, and a good dose of passion and curiosity. The goal is to control, eliminate vaccine-preventable disease, but also vaccine-induced disease. The progress has been terrific in some developed countries, I would say in most developed countries, but there are still sizable gaps in resource-limited settings. On this diagram, you are familiar with the pre-marketing phase of development, of vaccine development, where safety data are collected during animal studies, pharmacologic knowledge, and also, of course, during the clinical development of phase 1 to phase 3. Then, once you have achieved successful clinical development, you require, you go for market authorization, and then starts a new phase that is post-marketing with 2 parts. Part that we call passive surveillance, that is by spontaneous reporting schemes, spontaneous reports, and active surveillance through clinical trials phase 4, observational studies, case control cohort studies, case series, record linkage methods, and specifically designed safety studies and registry and large sample safety studies. We will focus on the post-marketing safety collection. The collection of AEA5 in post-licensure. What are the source of data? There are the spontaneous reports, the literature reports, the reports of vaccine registries, the reports from the internet websites, from post-licensure studies, and from the licensor or licensee. So over the life cycle data from continuous safety monitoring strategies provide complementary insights in vaccine characteristics. Important are the definitions. They are absolutely key to know what we are talking about. The first is to define vaccine reaction, untoward biologic effect caused by the vaccine, adverse events following immunizations, untoward event post-vaccine administration, vaccine reaction, programmatic error, coincidental or unrelated or unknown, and importantly, the adverse events of special interest, any untoward medical event that follow immunizations and that does not necessarily have a causal relationship with the usage of the vaccine. The adverse event may be any unfavorable or intended sign, abnormal laboratory findings, symptom or disease that may lead to the detection of signal. I will come back on signal. So what is the system the global system to collect this data that would be generated by the method that I exposed before? For more than 30 years, the Uppsala Monitoring Center, UMC, has provided the scientific leadership and operational support to the WHO program for international drug monitoring, what we call the WHO PIDM. And UMC is the maintenance organization for VGBase, which is the WHO international database for suspected adverse drug reactions, and for the WHO PIDM. VGFlow is a national data management system that's been offered since 2001, 20 years ago. And VGLease has evolved from being only a global data reference to now a powerful search and analysis tool for national, regional, and global data. So here you have a series of figures with here the flow of data from e-reporting, primary reporter, industry, or mobile safety app interoperability managed by the MHRA, for example, in the UK, and the VGFlow and VGBase. So the data that I entered in VGFlow go to VGBase, and from VGBase, they can also go to VGLease for national, regional, and global signal detection and signal management. There are 93 countries that are using VGFlow globally, and you have also 142 members of the WHO PIDM, 29 associate members, that covers more than 95% of the world population, and all can have access to the VGBase data. So in terms of methodology, passive surveillance, how do we define passive surveillance? This is a spontaneous reporting of adverse events following immunizations by healthcare providers, immunization providers, consumers, and by other sources to the appropriate level in each country, depending on its national pharmacovigilance reporting system that is usually located at the national regulatory authority, and to the marketing authorization holder. It is a relatively inexpensive strategy to cover large areas, but data quality and timeliness are difficult to control. Here you have the strengths of spontaneous reporting. I will go quickly. They cover all medicinal products and vaccines and the whole population. They have the ability to detect AFI that are rare, unexpected, unknown, and clinically relevant and serious. They also allow the early signal function, signal generation function, the tip of the iceberg. They are inexpensive and less labor-intensive strategy to cover a large population. There may be some ways to enhance this passive surveillance by public information, to encourage also through campaigns, encourage and facilitate reporting in specific situations, by systematic stimulation of vaccine administrators, by stimulation of strategies focusing on EFI, and resources and efforts more effective by limiting stimulated reporting to a few sentinel sites. And these methods to enhance passive surveillance can be achieved through telephone, online reporting, apps, systematic stimulation via reminders, personal visits, important to motivate, and additional training to healthcare providers. The weaknesses are there is no direct information on incidents that is collected. Reporting rates are not stable over time, with a risk of underreporting, and the underreporting decreases the sensitivity and sensitive to selective reporting. It is not representative, there is a bias, and the clinical information often too limited in terms of quality and quantity. And the case evaluation of causality assessment is questionable. There is no control group, and the general signal can be tested. Poor case identification, and possibility also to fake AFI. I must also add, it's not on the slide, that in some settings, in particular the poor settings, the healthcare workers, the health officers are overwhelmed by so many tasks that adding passive surveillance to their task is not often a priority for them. The factors that are influencing reporting are listed here. The main things that are important is the topics that are circled here. The main cause related to underreporting were ignorance, insecurity, and indifference. These are factors that can be addressed by programs of training, education, and motivation with the physicians and in hospitals and health workers in any healthcare facilities. Different is the active surveillance method. This is a data collection system that seeks to ascertain as completely as possible the number of adverse events following immunizations in a given population by continuous organized process. It provides the most accurate and timely information, but it is an expensive strategy. The type of data needed need to be complete, reliable, unbiased on vaccination. The outcome data on health events are also absolutely key to be collected, as listed below, and the population demographic data to define the population at risk and the individual level and medical information. The active surveillance are the classic, the cohort and case control studies are the classic post-licensure methods for investigating risk in this active surveillance methodological approach. But you have also, you can see that you have also clinical trial, phase 4 and post-authorization safety studies. But let's focus on these cohort event studies. So the advantage of the cohort studies is that this is a prospective approach from cause to effect, and respective studies using data set collected with other purposes, which allows a large linked database usage. A relative risk directly computable, and assessment of multiple outcomes, standardized observation, and transparent analysis. The problems are the selection of the comparator group. Studies must be very large for rare AFI, and AFI with long latency intervals are more complicated to catch. They are time-consuming and lost to follow up participants also may account also for some difficulties. And it also needs a considerable logistics, and these studies are costly. Here's an example from research limited setting where you have example of a comparison of the PCV10 from a 2-dose vial without preservative compared to the pentavalent. And this cohort study concluded that there was no difference in terms of AFI between the two vaccines. Case control studies are the second method that is often used. The advantage is that they allow to detect rare events with long latency intervals. They are fast and inexpensive. There is no ethical issues, and many etiological factors at the same time can be analyzed. The problems are they are prone to bias by the selection of cases, selection of controls, and the exposure assessment. The second set of problems is the retrospective approach, and the relative risk and attributable risk are not directly computable. Here is an example of a case control study for the study of intussusception risk and health benefits of a rotavirus vaccine in Mexico and Brazil. I can go to the article. I will go relatively quickly on this, the case series that are self-controlled. This is a methodology that is using only data on cases, comparison not made between individuals, and the risk estimation only within individuals. Here is an example of post-authorization safety surveillance of a liquid pentavalent vaccine in Guatemalan children. The advantages of this case series is that they are self-controlled designs, expand epi research capabilities. They are best suited for relatively acute events and transient exposure with defined periods of risk exposure. They allow monitoring of defined AEFIs in timely and cost-effective manner. They are active surveillance in resource-limited settings where unexposed populations is difficult to find. The problems is that inclusion of pre-vaccination time periods may bias the association between vaccine and events. Another method is the aggregate data without individual linkage, the observed versus expected, where the attributable rate is the difference between the observed rate and the background expected rate. And here is a fake example where you have this breakdown between observed and attributable, background expected and observed, and the attributable rate would be this light green box. Importantly, there is a critical role of background rate of possible adverse events in the assessment of COVID-19 vaccine safety. I will take this example because we are in an exceptional period where there has been, with the emergence of the COVID-19 and the pandemic, unprecedented efforts to develop vaccines have been consented worldwide. And all vaccines are now being introduced globally with inequity, as we know. We face the prospect of millions of people being vaccinated in multiple types of vaccines, many of which using new vaccine platforms. So since medical events have happened without vaccines, it will be important to know at what rate events occur in the background, so that when adverse events are identified with the vaccines, this background rate would be a frame of reference to compare the rates observed post-vaccination. This is absolutely key to better understand, in particular, adverse events of special interest. The background rates vary over time by geography, gender, socioeconomic status, and age groups. There are two key steps to post-introduction safety evaluation of COVID-19 vaccines: defining a dynamic list of adverse events of special interest, and establish a background rate for these AESIs. Some signals, as you may have heard, have been detected with different vaccines, and the data that I present here are not updated. They are updated regularly. There are flurries of papers in the literature. One that came very strongly following the AstraZeneca vaccination is the thrombocytopenia with thrombosis syndrome, TTS, which is rare, very rare, 1 in 100,000 vaccinated individuals, mostly young women, but not only. The capillary leak syndrome, so very rare, and some find it controversial. The Guillain-Barré syndrome, there is a suspicion, but there is no proven link. It needs considerably more data to say that the DBS is a signal post-COVID-19 vaccination. Similar observations have been observed with the J&J vaccine. We don't know for other vectors like the Gamaleya and the CanSino vaccines. Another signal that has emerged recently is post-RNA vaccines, the myocarditis, pericarditis. Here you have some tables of the characteristics of preliminary reports from the VAERS, which is the US system following RNA vaccinations. And the table on the left, below left, indicates clearly that there is, between expected and observed, a considerable higher number of observed events compared to the expected. Again, it illustrates well the importance of knowing the background information. On the right side, you have, and again, these data are from May 29, so I didn't have time to update the slide to have more recent data, but the trend was already noticed that post the first dose, you did you did have some myocarditis, pericarditis incidents, but that was very flagrant and obvious after the second dose, both for Moderna and both for Pfizer-BioNTech. So this is what we call a signal. And you can see that the numbers to come to this analysis are quite consequent when you look at the dose administered. So in summary, for this myocarditis, pericarditis, the initial safety findings from Pfizer-BioNTech, a 12 to 15-year-old for vaccine are safe from the VRI surveillance are consistent with results from pre-authorization clinical trials. The preliminary findings suggest that there is a median age of reported patients with younger, with median time to symptoms onset shorter among those who developed symptoms after dose 2 versus dose 1, predominance of male patients in younger age groups, observed reports superior to expected cases, as we have seen, and limited outcome data suggests most patients had full recovery of symptoms. So these are a series of reference that you can consult the European Network of Central Centers for Pharmacoepidemiology and Pharmacovigilance, Council for International Organizations, the CIOMS, the WHO, EMEA, and the Benefit Risk Assessment of Vaccines, and the Brighton collaboration or safety platform for emergency vaccines, and some articles are absolutely key. I would like to acknowledge my colleagues of the COVAX Vaccine Safety Working Group, in particular Bob Chen, former CDC and now scientific director of the Brighton collaboration, Steve Black, also Brighton collaboration, Katrina Hartmann, and all my colleagues below. It will be too long to list, to cite. Kamsahamnida, thank you, and stay safe.
Thanks, Jean-Louis. So unfortunately, Jean-Louis could not join us for the live Q&A session. And although we did not really plan to have this, we are thinking about how do we get this, some of the interesting questions that we can get answers from the speakers and get back to you. So we'll announce that if we have some plan about that. So now we move to the last lecture of today's program, and it's my pleasure to invite Professor Pauline Paterson from London School of Hygiene and Tropical Medicine. And, now we are seeing so many issues around vaccine hesitancy. It looks like there is a lot of miscommunication, misinformation, and she's going to talk about the vaccine communication and vaccine hesitancy.
Hi, my name is Pauline Paterson. I'm an assistant professor at the London School of Hygiene and Tropical Medicine, and I'm co-director of the Vaccine Confidence Project in London. And today I'm going to be talking to you about vaccine communication and vaccine hesitancy. So the team at the Vaccine Confidence Project, we conduct global research on vaccine confidence, examining local and global dynamics which influence vaccine decision-making. So our main purpose really is to understand why people aren't vaccinating. And in our team, you can see here the list of the team members in our research team, and we also have PhD students and MSc students that do their research with us. We're an international team of experts in anthropology, epidemiology, statistics, and political science. And in terms of what we try and examine, we don't just look at confidence in vaccines, but also confidence in providers and confidence in systems. And the aim is to detect early signals of concerns about vaccines to get them addressed before they amplify. After clean water, vaccination is the most effective public health intervention in the world for saving lives and promoting good health. However, while most people vaccinate, some people, groups, individuals, delay or refuse vaccines. The World Health Organization definition for vaccine hesitancy is a delay in acceptance or refusal of vaccines despite availability of vaccination services. It's complex and context specific, varying across time, place, and vaccines. And it includes factors such as complacency, such as a lack of perceived need or value for a vaccine, convenience in terms of access to the vaccine, in terms of cost, but also in terms of how easy is it to book an appointment, how far away is the vaccination center, and confidence. So the level of trust, not just in the vaccine, but also in the provider or the process. Now, in the old, way back when, we used to, and also sometimes in the media, you'll see this, that people refer to the anti-vax and the pro-vax. But actually, there's more of a continuum. So there are those at the small minority that do refuse all vaccines, as you can see at the top of this pyramid. But then there are those that delay some vaccines, but not others. And then there are the group that are more hesitant. They comply, but they have questions. And those that accept cautiously, they've got a few concerns. And then the majority of people do accept vaccines. And without question, they just go ahead and get vaccinated because it's recommended for them. I wanted to give you a few examples. So here's an example with MMR vaccine, measles, mumps, rubella vaccine in England, where there was an article published in The Lancet by Andrew Wakefield and colleagues, and this was published in 1998. And as a result of the article, so this article suggested a link between the vaccine and autism, and there was media coverage as well. So it wasn't just the article. The general public don't read Lancet articles generally. So there was a lot of media. The Prime Minister at the time also would not say whether their son was vaccinated with MMR vaccine. And so, as you can see here, the graph shows that coverage in England went down from 91.8% to just below 80%. And what we want to show here as well is that concerns can take time to develop in individuals' minds, but in groups, and to spread as well. It can take some time, and also it can take a while for coverage to go down, and it can also take time for coverage to then increase again. So the Lancet article was partially retracted, then it was fully retracted. There were a lot of research showing that there was no link between MMR vaccine and autism, and there was media against Wakefield. He was struck off the medical register. There was a catch-up campaign and communication to rebuild trust, but it took a long time to get coverage back up to, well, so this was in 2014 in this graph 15, to just above 92%. And with measles, because it's highly infectious, you need coverage to be above 95% to not get outbreaks. So this shows that perceptions of risk, concerns around vaccine safety can cause dips in coverage, and it can take a while to rebuild that trust and get coverage back up again. This is another example with measles vaccine. There was a study by ENVS in France, as there was an outbreak of measles in 2010, 2011. And so this study looked into the reasons why parents weren't vaccinating their children with measles-containing vaccine. And the reasons why were either they didn't perceive measles as a serious disease. The parents themselves back then had, when they were children, just got measles. They didn't have a vaccine. So they thought, well, I had measles, I was fine, so my child will be fine. There was this perception of the vaccine risk being greater than the disease risk. And also some communities have public distrust of authorities and medicines and or pharmaceutical companies. And some groups of people believe in homeopathy. So they would prefer to have the disease than get vaccinated. The SAGE working group created these determinants of vaccine hesitancy that, just to go into a bit more depth than the 3C model. So here, as you can see, there are contextual influences. These are influences arising due to historic, sociocultural, environmental, health system, institutional, economic, or political factors. There are individual social group influences. These influences arising from personal perception of the vaccine or influences of the social peer environment. Vaccination specific issues that are directly related to the vaccine or vaccination. And just to show that safety concerns are just one of the many determinants of vaccine hesitancy. And there's safety concerns, so you can see in the vaccination specific issues, there's the risk benefit, the scientifically based risk benefit. But in the individual social group influences, there's the perceived, the heuristics risk benefit as well. Exploring COVID vaccine hesitancy. There's been quite a few studies already looking at reasons why different members of the population are vaccinating or not, and exploring whether people would accept a vaccine should one become available and be recommended. This was in the earlier days to when there was a vaccine available, why people weren't vaccinating. And this just shows you the range of vaccine acceptance in these different countries. And if there's a range in a country, it means there's a couple of studies that have come up with different results. So this will really depend on when people were asked, who was asked, how they were asked, like the question itself, but also what options they have in their answer. Whether, it's a yes, in the question, for example, if it's recommended to you and available and safe, that would be one question. But in another country, maybe they didn't add the and safe, or they didn't say it was recommended for your group. So there are little nuances that will affect whether people would respond to whether they'd be accepting of the vaccine or not. The study by Lazarus surveyed in, I think it was 19 countries. So then you can compare for that a bit more readily what the acceptance rates are. We conducted a study. I forgot to mention in introductions, I'm part of the NIHR Health Protection Research Unit in Immunization at LSHTM. So we conducted a study in parents and guardians of young children. And the primary aim of our study was to explore how their experience of vaccinating their children against childhood vaccinations and how that went, and whether they did, or whether they delayed during the first lockdown, so last spring in England. And we also asked them whether they'd be willing to accept a COVID vaccine. So we asked, if a new COVID vaccine became available, would you accept the vaccine? And 90% responded yes. And the main reasons they said they would accept one was to protect themselves, their family, others, to stay safe for their children, to stop the need for social distancing. And the main reasons for not accepting the vaccine were concerns about safety, that it was a new vaccine. So this was spring last year and it wasn't available then. The process has been rushed. There's not enough evidence. And some, it wasn't that they were concerned. It was more that they weren't in an at-risk group. So they thought, let's give the vaccine to those that are in at-risk groups. I don't need it. And also there were some concerns about a possible lack of effectiveness. Some of the key findings were that Black, Asian, Chinese, mixed, or other ethnicity respondents were almost 3 times more likely to reject a vaccine for themselves and their children than the white, British, white, Irish, and white other respondents. And also lower income households were more likely to reject a COVID vaccine. So the main purpose of our study was to look at views and experience of accessing routine childhood vaccinations in England. And we found that almost 1 in 4 had experienced issues or difficulties. So sometimes it wasn't clear whether vaccinations were still happening. They might have booked an appointment for their child, but then they got a routine text message from their GP, from their doctor saying, don't come into the surgery, everything's closed. But it was just a routine text message, so it was confusing. There was a lack of clarity around vaccination services, whether they were an essential service or not, and whether they were happening, and also what measures had been put in place to keep patients safe. And some parents, they said, well, we're not going out. I interviewed one mother. She said, my daughter's 2 months old. She hasn't met my family. She hasn't met my parents. We haven't seen anyone. Why should I take her out and risk? Because in the UK as well, in the GP service, sick people go to the GP as well. So they wouldn't want to take their child where there might be sick people. Yeah, I just wanted to show this because with the outbreak, with the pandemic, comes as well misinformation online. And every outbreak is accompanied by a wave of information, and with that information comes misinformation, rumors. Dr. Tedros Adhanom, the Director General of WHO, warned against an infodemic of rumors and misinformation that needs to be fought alongside the pandemic. And with the internet and social media, individuals are more informed and empowered than ever before. But with this overload of information, also, as I was saying, there's misinformation and conflicting information. You can pretty much go online and find anything you want to find. If you have a specific concern, you will find that concern out there and other people with the same concern. And as I was showing with the MMR case study in England, and it took a while for coverage to go down, with social media, with the internet, concerns can spread quickly and far. And we did a study at LSHTM, but with collaborators, looking at online misinformation around COVID vaccine in the US and in the UK. The references there if you're interested. And it did show that exposing people to misinformation around the vaccine did reduce people's intentions on vaccinating. This I wanted to show at the Vaccine Confidence Project. We developed a vaccine confidence index to track public trust in vaccines around the globe. And there were 4 key components. Overall, I think vaccines are important, vaccines are safe, vaccines are effective, and vaccines are compatible with my religious beliefs. And the answer was on a Likert scale from strongly agree to strongly disagree. And this was the largest study to date. And as you can see here, so this is for vaccines are safe, and the map on the left is for 2015, and the map on the right is for 2018, with the heat map of respondents who strongly agree that vaccines are safe. So as you can see, the confidence varies widely between countries, and in different countries it's either gone up or down. But in 2015, the European region had the highest negative responses for vaccine importance, safety, and effectiveness. And although confidence has remained low in the European region, it is improving in some member states. So it was very low in France, but it has been improving. It's also been improving in Italy, Ireland, and Finland. And this just gives, so it's a very simple metric, but you can apply it widely. This was in 149 countries, and it can give you an indication of what's happening. And if there's a need for a deeper dive to try and figure out, okay, there's an issue, or if you're introducing a new vaccine, such as with the COVID vaccine, are there issues already in this population? Is there distrust already in vaccine safety, for example? This decision-making model visualizes selected determinants of vaccine decision-making, particularly relevant in predicting the vaccine decision. So there's information such as campaigns, education, recommendations, media, and then there are these risk perceptions. So the risk of the infection and the risk of a vaccine adverse event. And then there are these modifying factors: attitude, identity, injunctive norms, what you should do, descriptive norms, what others do, habits. If you've vaccinated in the past, you're more likely to vaccinate again in the future. And then these barriers, these convenience barriers, costs, time, and effort. And then someone might be intending to vaccinate, but then there's an extra leap to acting and going ahead and getting vaccinated or getting your child vaccinated, for example. Now for communication on immunization to be effective, trust and credibility in public health organizations is key. Transparency about actions and decisions is essential in maintaining public trust. It allows the general public to understand the information gathering process as well as the risk assessment and decision-making processes associated with immunization issues. Information should be easily accessible, accurate, consistent, timely information. It's really important that when there is new information to share it, or even a lack of information to share that there is information coming, for example. People fear the unknown, and they may have a tendency to exaggerate their fears if there is a lack of information. I wanted to write about dialogue as well here, because it's not just about communicating out, and it's not just assuming that if people aren't vaccinating, they're not educated enough. It's really important to have a discussion to understand how people are feeling about vaccination, about the process, about just engage the public and stakeholders. So it's really important to have dialogue, because it also, you get informed, you find out how people are thinking, and then it can help inform your messaging as well. But it can build trust and share knowledge and ensure mutual understanding. So these factors alone will not ensure widespread trust. The public must see that competent and well-informed decisions are being made based on evidence. And which channels should you use to communicate about vaccines? Well, there's a number of channels, and these are just to give you a flavor of them. But you really need to figure out who you're trying to target when you are communicating and where they go. So you need to be where they are. So if they're online, you need to be online as well. If they're talking to their religious community leaders, it's really important to talk to them as well. Healthcare workers are the most trusted source of information, so working with them too. And it's really important not just to go to one of these sources, but to go to a variety of sources as well and share that information, because it's really key that the messages are consistent throughout the channels too. Who do people turn to? This was a study looking at transparency, but also at trust, looking at medicines information in these 6 countries. And they found that, so in the gray is where people go to find information about medicine. So you can see they go to the GP, pharmacy, the local hospital, the internet. But if you look at the black, which is where people trust, people don't tend to trust the internet, although they go there. They trust more the GP, the pharmacy, the local hospital. I wanted to give an example of HPV vaccine because it's a different kind of vaccine, and because it's given in teenagers. And so that as a result of that, there's also other issues and other concerns. So the concerns around the HPV vaccines, this is a human papillomavirus vaccine, which prevents cervical cancer and a number of other cancers as well, is there's been different concerns. So for example, in Japan and in Denmark, there were issues around POTS, CRPS. So POTS is postural tachycardia syndrome. So these fits in India. There was a video shown on YouTube of a teenage girl after being vaccinated having seizures on a screen on YouTube, and this spread massively, and there were concerns around that. In India, there were concerns that there were clinical trials. There were demonstration projects, but it was a bit unclear. There were issues around ethics there and discrimination, but there were also concerns around safety around the vaccine. In the US, there were more concerns about sexuality because HPV is contracted sexually. So there were concerns from parents that if they allowed their daughter to get vaccinated, that they were allowing other things to go on. There were also issues around mandatory vaccines. In Colombia, there were concerns because there was episodes of mass fainting of young girls during their HPV vaccine programs, so there were concerns around that. And then in terms of communicating, so Public Health England and NHS England, they actually spoke to parents and they did focus groups. And although Public Health England say they didn't really like this, well, it's not that they didn't like this logo, but they had a number of logos that they designed, and then they asked parents, and and the teenagers what logo they liked. And this one they really liked. So they went with that for one of their campaigns, and they're making it young and kind of speaking to teenagers. And in the US, there's this website on the CDC which explains different ways for healthcare providers to discuss HPV vaccine, and if parents have questions or concerns, how to respond to this. ECDC also has a great resource for communicating around and addressing misconceptions. So what's really key here is scientific research in psychology has shown that addressing misconceptions is difficult. Mere reading about a myth, even its refutation, can strengthen the myth. So you've got to be really careful when you are communicating about myths and not use strong language, because then the reader can come out with a higher risk perception as well. And just to summarize, I'm sorry, it's a short lecture, but hopefully it'll give you a flavor of vaccine hesitancy and vaccine communication. So while most people vaccinate, some groups or individuals delay or refuse vaccines. This can lead to vaccine refusals and disease outbreaks. Research has shown vaccine hesitancy is complex. It really does vary by vaccine, by time, by context, by geographical region, by subpopulation. But in addressing vaccine hesitancy, it's really key to identify those groups that aren't vaccinating, and then to figure out why they're not vaccinating. So to talk to them, to conduct studies, either surveys or in-depth interviews or focus groups or media monitoring, and figure out why people are not vaccinating, and then to address those reasons. Thank you so much.
Thank you so much, Professor Paterson. So actually, Pauline is here with us for the live Q&A. Can we bring her? Yes, Pauline, as always, quite informative, and especially with the COVID-19 vaccine, there is a lot of hesitancy, there is a lot of miscommunication, and as you can imagine, there are a lot of questions that are coming in the Q&A session for us. We have selected few questions for you to answer, and I will begin with one very interesting question that I felt was, it will be quite interesting to see how you react to that. The question is, does hesitation to receive a specific brand of vaccine that fails to protect against certain variants of COVID-19 count as vaccine hesitancy?
Yes, thank you so much for having me today. I apologize, the video is a bit slow with the internet connection, but hopefully you hear me okay. So yes, I think it would be defined as vaccine hesitancy, because if that is a vaccine that's available to that person and they're thinking they might not want the vaccine, then I would consider that vaccine hesitancy, yes.
Perfect. Thank you. So second question is, how can vaccine hesitancy be taken into account when you are planning for a vaccination campaign? And this is a generalized question, not specifically for COVID-19.
Thank you. Yeah, it's really important, even from before a vaccine program starts, thinking about whether the public want the vaccine, will accept the vaccine, and what their hesitancy is. So what the reasons might be for not vaccinating. And also, I was answering in some of the other questions, it's really important to look globally to see, like, how is your country comparing to other countries, but also, the countries, the regions, subnationally, are there specific groups that you need to target as well? And it's really important in terms of communication to really modify it based on who you're talking to, that not just having a generic vaccine campaign. So, and it's really important as well to engage the public and not just shout louder, not just communicate more, not just educate. It's not a question of a lack of education. It's really important to figure out, just keep an eye out on vaccine coverage figures. Generally, when we work with public health officials and program experts, they know where there's an issue and they're working towards it. But it's really important to figure out, well, what are those reasons? Because it's not necessarily that the public don't want it. Maybe they had trouble accessing it. Maybe they don't know where to go. Just figuring out what the main reasons are for that under the lack of coverage in certain populations, and really being careful not to stigmatize populations as well that are choosing not to vaccinate.
Thank you. The next question is, do you think that compulsory vaccination policy will make things worse or better?
Yeah, I tried to answer some of the questions, but some of them are so complex it's quite difficult to answer in a couple of sentences. But my personal view is mandatory vaccination isn't the answer. It's really important. Figure out why people aren't vaccinating. And sometimes by the time you've put the processes in place to make it accessible, make vaccines accessible to, let's say, some countries, make the vaccines accessible to get to the point where they can mandate it. They don't need to mandate it because they've made the vaccine so accessible that they don't need to mandate. But I think mandatory vaccination is an issue. There have been studies in the US showing that, for example, mandatory vaccination of flu for healthcare workers has shown an increase in uptake, but you also need to look at your context. If your coverage is high already, it's not worth mandating. I mean, there are some cases where it might make sense, and also there's a lot of different kind of ways of describing what is mandatory vaccination. So in some countries, you can't get childcare benefits if you don't vaccinate your child, or you can't send them to school. I think that's not the answer. If a parent is not vaccinating their child, not sending them to school is not the answer. Like, you really need to figure out what are the main reasons and kind of address those underlying reasons.
Perfect. So I think we will take a few more questions since we have the time. The next question is, did the initial controversy and eventual suspicions about the COVID-19 outbreak, the outbreak itself, do you think that affected the acceptance of vaccine in the UK and elsewhere in the world, as per your knowledge?
Yeah, so what we did is we did a study on misinformation around vaccines in the US and in the UK, and we found that exposure to misinformation around COVID vaccine does reduce intent to vaccinate. So there have been studies showing that although people go to the internet, they don't necessarily trust it. This study did show that it does have an impact. And we're trying to untie this. We've got a UNICEF project at the moment where we're trying to figure out, like, do attitudes change based on what people are seeing online? Because the issue is there is so much information online that pretty much any kind of, if you're worried about something, you search for that online, you're generally going to worry more. But what we're trying to unpack is, where do people go? Because we need to trust people, because generally people will go online and search information that reinforces their pre-existing views. So people that are pro-vaccine but have some questions will go to CDC, WHO, like Public Health England. They'll go to reliable sources, whereas people who are more skeptical will go to sources that will agree with them. So this is something that really we're trying to unpick as well, is to figure out exactly where people are going and looking at how concerns spread globally online, and the social networks as well we're looking into.
Thank you so much. So in that line, probably we can take this last question. What are the most effective ways to counter COVID fake news? And then the question continue. Should scientists collaborate with the media people to address the vaccine hesitancy brought about by the inappropriate news? Should there be guidelines in place for media persons to follow in order to communicate scientific information effectively? Again, there are three questions in one question probably, but
yes, that's fine. I'll start with the last one. So in the UK, we have a science media centre where the scientists talk to the media and try and train the media in their science, and the media centre trains the scientists in how to communicate as well. And I think that's really important to have that dialogue with the media, because the media can create controversial kind of headings and cause fear and cause concern. So you have to be careful there. In terms of addressing fake news and misinformation, the pro-vaccine movement is less nimble and less responsive than the anti-vax or the vaccine critical people online. So that can be an issue. And there can be a lot of misinformation online. So it's really important to figure out where people are, so which social media platforms people are going to, and to be there as well. And although it can be a bit daunting for vaccine experts to tweet and to be on Facebook, and also sometimes you feel like it's a losing battle, because people who are very anti-vaccine, you can spend hours and hours discussing and you're not going to change their mind. But it's really important to be in these spaces where those people who are kind of in the middle, who are looking at the anti-vaccine information, need that pro-vaccine information as well. There are some collaborative efforts between platforms and public health agencies to detect misinformation and disinformation. Disinformation is when misinformation is spread purposefully. And there are efforts, social media companies are reducing and removing content that is disinformation. And there are efforts to counter misinformation as well, and to direct users to official sources. So you see this on Twitter, where you're directed to the NHS, for example, in the UK, if you search for a specific topic, and banning ads. But it's really important, and there are efforts too to improve the public's media, digital health literacy. So Facebook had a misinformation vaccine. So if you teach the public how to spot fake news and to look for the source and to think, okay, why is this being shown to me here? Like, can I trust it? What's the website? That sort of thing. Then these initiatives can help with slowing the spread of misinformation and disinformation, and hopefully reduce vaccine hesitancy as well.
Perfect. I mean, there are a lot of questions, if you can see in the Q&A, but since we have limitation of time, again, thank you so much, Pauline. It's always a pleasure to have you in our vaccinology course. So with that, we are coming to the end of today's session. It was day 4. We had good participation from all of you. Again, thank you so much to all the panelists, to all the moderators, to all the speakers, as well as the participants. So this is the end of day 4, and then we look forward to welcome you again tomorrow for the last day of this year's vaccinology course. Thank you so much. Stay safe. Good evening. Good afternoon. Bye-bye.